Hello, good morning to everyone. My name is Ernesto Llamas. I'm a postdoctoral researcher at the University of Cologne. And today I'm going to talk about fighting neurodegenerative diseases with the power of plants, an innovative approach that we have been working in the last few years. So first, I would like to introduce my team. It's formed by David Vilches, that is working in the Cluster of Excellence of Aging Research, and Dr. Alga Zuccaro, that she's working in the CEPLAS in the Cluster of Excellence of Plant Research. So this protein involves biomedicine and plants, and believe me, to find a collaboration between these two sides, it was so difficult. I sent thousands of emails, but at the end I managed to get the collaboration between CEPLAS and CICAT. So I'm a postdoc working there, so now we are trying to spin off this idea from these two clusters of excellences. And I want to talk today about a certain type of diseases that are called the polyglutamine diseases. So they are 9 different polyglutamine diseases, and they don't have a cure. For example, Machado-Joseph disease and some spinocerebellar ataxias, and of course, Huntington's disease. All of these diseases, they have something in characteristic. So they have a mutation in the gene that codifies for the proteins that expands the polyQ region, a certain repetition of amino acids that makes the protein to become unstable. I always use an example for explaining this problem in the proteins. So when you have a protein that is not mutated, it's correctly folded, like a paper plane that it was already mentioned before in the presentation. So the protein is active because it's correctly folded, of course. It's flying, right? But when you have the mutation in the gene of these proteins, then you have a protein that is incorrectly misfolded, and the activity is compromised, and the paper plane cannot fly that optimal like the good ones. So these accumulations of paper planes in the neurons is causing these toxic effects and death in neurons. So I'm going to give more details about Huntington disease. So affects 1 in every 10,000 people. And that means like around worldwide is 1 million people affected right now. And it can be developed, if you have the mutations, it's an inherited disease. You have the mutation, you can develop at any time point of your life. And it's, when you get the symptoms, normally it's like between 30 and 40 years old, but sometimes people, even with 20 years old, they develop the symptoms. And actually, this is a very devastating disease because when you start the symptoms, having the symptoms, then your life expectancy is around 15 to 20 years. And even though that the current efforts and research in the last years have been done by many scientists around the world, we still do not have a cure for these diseases. And also, we don't have a cure for the polyQ diseases. But I'm a plant biologist by training and a plant molecular biologist, and I know that plants can live longer than us. So the sequoia tree can live thousands of years, for example, and the human average lifespan is 80 years old, for example. So I know, and some scientists have been doing research about why plants, they don't have this kind of problems with protein aggregation, with these paper planes that are aggregated. So we went into detail to analyze this process, why they are resistant to this kind of protein aggregation. And what we did, we put the mutant gene, human gene, into plants to see what happened with the plants. So we were curious about what happened when you introduce these toxic proteins into plant. And this is the result. So it's not spectacular. The plants with the Huntington protein or the polyQ extended protein, they look like the control plants. So we were surprised that the plants are able to resist this protein aggregation. So then we did more molecular studies to characterize what was special in the plant that was avoiding the aggregation of this human toxic protein, and we discovered the key in the chloroplast. A certain protein is binding to this and just clearing the aggregates and avoiding the toxic effects. But you will say, so humans, we don't have chloroplast. Of course, we don't have, and also the mice or the research models of disease, they don't have chloroplast. So what we did, we copied this protein that we found in plants with synthetic biology, and we put it in these models of research in human cells and C. elegans that they are affected by the disease, so they have these polyQ proteins in the neurons. And when we put the protein, so we were able to clear the aggregates in this model of research. And the C. elegans were normally with this disease, they cannot move, and we restore motility after putting this plant-based protein. So now this is the solution that we want to offer in the future for the market. So we want to create an enzyme-based drug to treat polyQ diseases. This is important, not only Huntington disease, but the whole families of polyQ diseases, that there are 9 in total. So it's also designed to be injected into the bloodstream, and it will be modified to cross the the blood-brain barrier that normally is a limitation for this kind of drugs to pass through the brain. So now I would like to talk about the market size of this treatment of the Huntington disease. So it was estimated that the market size in 2022 was $380 million, and it's expected the compound annual growth rate to be increasing 23%. And we don't have any data about the other 8 polyQ diseases. So we expect that the market size of this possible treatment is going to be way bigger. So what about our competitors and other strategies that have been developed about trying to cure Huntington's disease? So Roche partnered with Ionis Pharmaceuticals. They stopped clinical trials in phase 3 because of adverse effects, and something similar happened with Wave Life Science and Takeda. And there are other companies also working in solutions and trying this technology that they are using gene silencing space drugs. So basically what they are doing, they are trying to silence the expression of the mutant Huntingtin, so as you can see in the scheme. In my case, we are using different approach. So we are using an enzyme-based drug. So that means that enzymes normally are more stable than nucleic acids that using the other technology. So that would allow to administer in a different way in comparison with these drugs. And this enzyme is going to be designed to specifically clear the misfolded proteins and leave the other proteins that are correctly folded alone. So now I would like to just to explain where are we now. So we are basically transferring now our academic knowledge, and the objective is at the end to generate a spin-off from the University of Cologne. So now we filled up a patent application. So we got recently funding from the German Ministry of Education and Research. They go by initial program. And actually, I'm pretty happy about to get this because this is one of the programs that BioNTech, at the beginning, they got it. So BioNTech is like the example that I want to follow to develop this kind of disruptive and different therapies. And if this first funding phase is successful, then I will be able to have more money to achieve also freedom to operate and to spin off the company. So that's why I'm very happy to be here, because I need the help from the audience and the help from the jury and the help here, because right now I'm recruiting currently a PhD student, so the position is right now open, so it's just three weeks more to apply. So if you are interested, please come to me. And also we are negotiating now already with some investors. It's a group of longevity therapeutic collective, because now they are interested in aging research. And I'm looking for drug experts, discovery, business, co-founders, team members, and of course in the future some investors. And with this, I leave you here my contact, and I thank you for your attention.